Welcome to the deep dive, where we sift through the noise and pull out the crucial, most surprising insights just for you.
hmm
Today, we're diving into a topic that might seem a bit specific, Seneca Valley Virus or SVV, but trust me, it has profound ripple effects.
It really does.
Our mission is to truly understand this enigmatic virus. It's causing, well, big headaches in the swine industry.
hmm
And often not because of the direct harm, but because of what it looks like, its ability to mimic other far worse diseases.
Right.
We've got a stack of sources here, foundational stuff, latest molecular research to give you the complete picture.
And that mimicry you mentioned, that's precisely why SVV is so critical for you to understand.
Yeah.
The pathogens that can be confused with really serious diseases, they pose this unique kind of persistent challenge.
Not just for animal health.
No, not at all. For global trade, economic stability, everything. Getting to grips with this master of disguise is really key to seeing why this deep dive matters.
Okay, let's unpack this then. We'll start with the basics. It mainly hits pigs, right?
Okay.
And causes these blisters, vesicles.
That's right, vesicles.
Yeah.
On the snout, mouth, sometimes the feet, distinct blisters.
And here's the core problem, isn't it? Those signs.
They are, clinically indistinguishable. That's the term we use.
Meaning they look exactly like...
Exactly like some really high consequence of dysmaphoses. Foreign animal diseases, FADs, we're talking foot and mouth disease, FMD, swine vesicular disease, SVD, vesicular stomatitis, VS.
Wow. Okay.
This incredible mimicry, this ability to look like the really bad stuff, that's SVV's main threat. It triggers just massive economic confusion, huge disruption.
A true imposter. So are there other clinical signs besides the blisters? Things you'd notice?
Oh yeah, absolutely. Beyond the vesicles, you'll typically see lameness. Pigs might be really reluctant to walk, sore feet.
Person.
They can also get a mild fever, go off their feet, anorexia, and just generally seem depressed, lethargic.
Okay. Now, I read something about a really counterintuitive twist. Its impact on the very youngest pigs. It's different.
Ah, yes. This is where the virus behaves quite differently and often tragically. So while older pigs get those skin lesions,
Right.
in neonatal piglets, like 0 to 7 days old, SVV can cause something called epidemic transient neonatal mortality, ETNL.
And the mortality rates there are high.
Shockingly high sometimes, anywhere from 30% up to, 70%.
Yeah.
But here's the really crucial part, the twist you mentioned.
Yeah.
These tiny piglets often die very suddenly without showing those blisters, those vesicular lesions you see in the older pigs.
Really? Why?
It seems to be a systemic, acute viral infection. It just overwhelms their systems before the skin signs can even develop. It really highlights this dramatic age-dependent pathology.
So different outcomes based purely on age.
Exactly. For adult pigs, thankfully, mortality is generally low.
Yeah.
But those lesions, they still cause significant production losses, lameness, things like that.
It's like two different diseases in one. Okay, let's shift focus. The virus itself, how's it classify? What is this thing?
Right. SVV is classified as a picornavirus.
Picorna, meaning
Yeah.
small RNA virus.
Exactly. Picornavirus. It's a non-enveloped, positive sense, single-stranded RNA genome, very small.
And that family, Picornaviridae, that's important.
Hugely significant. Because this same family includes FMD virus and swine vesicular disease virus.
Ah, okay. So that relationship explains the mimicry.
Precisely. It's fundamental biology. It was actually first spotted back in 2002, believe it or not,
Oh.
as a contaminant in cell cultures. Just, showed up unexpectedly. It was only later confirmed to actually cause disease in pigs.
So it was lurking for a while, given how widespread it is now. How does it actually transmit? Moving between pigs, between farms? Sounds like a tough challenge to control.
Yeah, it's tough. It have multiple routes. Direct contact, obviously, pig to pig or with their secretions.
Standard stuff.
Right. But also fomites. That's a big one. Contaminated equipment, vehicles, coveralls, boots, personnel moving between sites.
So biosecurity needs to be tight.
Absolutely. And then there's the oral fecal route, too. Contaminated feed, water, just surfaces in the environment.
And it hangs around.
Yes, it's pretty stable environmentally. It can persist in contaminated manure or bedding for quite a while, which just helps it keep spreading.
Okay, and I heard there's a particularly tricky aspect, something that makes it really hard to trace and contain. Carriers.
Ah, yes. This is a major headache for biosecurity. The persistent carriers.
Meaning pigs that look healthy?
Exactly. Clinically normal pigs, they can look perfectly fine, but they harbor the virus in their lymphoid tissues, especially the tonsils.
For how long?
For at least 60 days, possibly longer. They might not show any signs, but they can shed the virus intermittently.
Especially when stressed.
That's a key trigger. Things like transport, mixing pigs, farrowing. Stress can cause them to start shedding again. These silent spreaders act as this sort of hidden, cryptic reservoir.
Which makes control incredibly difficult.
Extremely. Because you're not just looking for visibly sick animals. Traditional biosecurity gets much harder.
Fascinating and deeply complicated. How does such a tiny virus cause so much trouble at the cellular level? What's it doing inside the pig cells?
Yeah, this is where its sophistication really shines, unfortunately. It's incredibly cunning, molecularly speaking. It uses multiple strategies to basically take over the host cell.
Like what?
Well, for one, it uses this unique mechanism, an internal ribosome entry site, or IRES, a type 4 IRES specifically.
Which lets it do what?
Think of it like a viral master key. It lets the virus hijack the host's ribosomal machinery, the cell's protein-making factories to make its own proteins.
Even after shutting down the host's normal production.
Exactly, even after it started shutting down the host's own translation. And what's really wild is its genetic mosaicism.
Meaning?
Parts of its genetic code, especially this IRES, structurally resemble bits from viruses in totally different families, like pestivirus or hep C virus.
So it's by road code, adapted.
It suggests it has, yeah. Incorporated genetic material over time, making it potentially more adaptable. It's quite clever.
So it's taking over the machinery. Is it also fighting the cell's defenses?
Oh, absolutely. It has a sophisticated, multi-pronged strategy just to dismantle the host's immune system from the inside.
How does it do that?
It uses specific viral proteins as molecular scissors. For example, its 3C prokeys, 3C Pro. It systematically cuts key signaling adapters in the interferon pathways.
Interferons are the alarm signals, right?
Precisely. Proteins like MAVS, TRIFE, PANK, cutting these basically severs the communication lines for the immune response. And other viral proteins pile on.
Okay.
The 2B protein also targets MAVS for degradation. The 2C protein directly blocks the cell from even transcribing its antiviral genes, the ISGs.
So it can't build new defenses.
Right. And then the 3A protein, it inhibits RNA interference, another key defense, by degrading a host protein called argonaute-2.
Wow. It's a full-on assault.
Yeah, it really is. And even one of its structural proteins, VP3, actively binds to and manipulates a host protein, HNRNPA2B1, to boost viral RNA translation while shutting down host protein production.
It sounds less like evasion and more like
Right.
complete cellular reprogramming.
That's a great way to put it. It is reprogramming the cell. And it manipulates it in other ways, too. For instance, apoptosis, programmed cell death.
The cell tries to sacrifice itself.
Normally, yes.
Heavy.
But SVV induces it late in its cycle. It seems the virus triggers it to help release new virus particles and spread.
So it uses the cell's suicide mechanism for its own ends.
It appears so. And one last thing, it reprograms cell metabolism, something called the Warburg effect, sometimes seen in cancer cells,
To do what?
to increase lactate production. And this lactate then actually dampens the immune response, seems to mess with the interaction between REI and MEVS, those key immune sensors. It makes the cell environment more welcoming for the virus.
That is genuinely wild. Such a tiny virus, such a complex strategy. Okay, let's shift gears. The economic impact. You said it's not always about pigs dying directly, so what's the real cost?
Right, this is the paradox of SVV, direct production losses. Actually, relatively low and sporadic, apart from that awful neonatal mortality, the ETNL. The real burden, the part that can cost, well, potentially billions, is the costly and disruptive indirect impact. And that all stems from its resemblance to FMD.
Foot and mouth. So how does that mimicry specifically hit the industry's wallet? Can you break down those indirect costs?
Sure. It sets off this immediate chain reaction. Any pig shows up with suspicious blifters.
Yeah.
It triggers a mandatory full FAD investigation immediately.
Which means?
Premises quarantined, stop movement orders for animals, maybe feed, maybe products. Processing plants might even have to shut down temporarily while tests are run.
And that just ripples outwards.
Exactly. It ripples through the highly integrated pork supply chain, hits producers, transporters, packers, everyone down the line. The disruption is immense.
And the burden on health agencies.
Huge.
Yeah.
Just look at the U.S. numbers. Between 2015 and 2020, the number of FAD investigations nearly quadrupled.
Wow.
And get this, 75% of those were triggered by suspected swine vesicular disease. Mostly SVV scares. That's a substantial and sustained burden on already stretched resources.
And then there's trade, I imagine.
Absolutely. The mere suspicion of FMD, even if it turns out to be SVV, creates immediate and severe market uncertainty. But the biggest fear, the nightmare scenario, is complacency.
Complacency. How?
That a real FMD outbreak happens and people initially dismiss it. Oh, it's probably just another SVA case.
And that delay.
Could be catastrophic. Tens of billions of dollars in losses for the U.S. economy alone. Immediate closure of basically all export markets. The stakes are incredibly high.
That sounds like an overwhelming problem. Is there any positive outcome, anything good that's come from this constant threat?
Well, interestingly, yes. In a way, SVA has acted as an economic catalyst.
How so?
The persistent threat, the constant FAD scares, it's essentially forced the industry to significantly improve its overall preparedness for all vesicular diseases.
So it's driven investment.
Exactly. Investment in better, faster diagnostics. A renewed, much more intense focus on really stringent biosecurity across the board. People realize the cost of prevention, even for SVV, is vastly less than the cost of responding to a real FMD emergency.
That's a really powerful silver lining, actually. Okay, so given the mimicry, how do they differentiate it quickly from FMD or the others? What are the diagnostic tools?
The gold standard right now is RT-PCR, specifically quantitative real-time RT-PCR, or RT-qPCR.
Detecting the viral RNA.
Yes, in samples like the fluid from the vesicles or swabs. To speed things up, diagnostic labs now use these multiplex RT-qPCR panels.
Testing for multiple viruses at once.
Exactly. One sample screened for SVA, FMDV, SVDV, VSV all at the same time gives you a rapid differential diagnosis. We also use serology ELISA tests, virus neutralization to look for past exposure for surveillance.
And what's on the horizon? Future diagnostics.
Oh, some really exciting stuff. Next generation sequencing, NGS, especially portable sequencers like the Oxford Nanopore Minion.
Portable, like on the farm.
Potentially, yeah. Rapid whole genome sequencing right there helps track viral evolution too, and maybe even more impactful soon. Research into rapid point-of-care tests.
Tests farmers can use themselves.
That's the goal. Things like RT-LAMP or RAA combined with visual readouts on lateral flow dipsticks, like a pregnancy test almost, results in maybe under an hour right on the farm.
That would be a total game changer for managing outbreaks.
Absolutely, for making quick decisions about quarantine and response.
So until those are widely available, what about prevention? What can farmers do now?
Well, since there's no specific treatment, and importantly, no commercial vaccines available yet.
No vaccines, okay.
Stringent biosecurity remains the cornerstone. That's really the main defense. Tightly controlling movement, animals, people, equipment. Thorough cleaning and disinfection is crucial. Luckily, disinfectants that work against FMDV generally work for SVV too.
That's helpful.
In managing those carrier animals we talked about, quarantine for incoming animals, maybe deliberate exposure of new breeding gilts during their acclimatization period to try and build some herd immunity in a controlled way.
That leads right to my next question.
And Vex?
Vaccines. You said none commercially available, but is there research? Hope on that front?
Definitely active research, yes, across several different promising platforms.
Your jazz.
Well, traditional inactivated vaccines, good safety, tend to give strong antibody responses. Live attenuated vaccines, they can give really robust immunity, but always that slight worry about them reverting back to virulence.
Right.
Then there are virus-like particle vaccines, VLPs. These are really interesting. They mimic the virus structure, so they're highly immunogenic, but contain no genetic material, so they're very safe.
Okay.
Also work on viral vector vaccines using another harmless virus to deliver SVV antigens. And even some early research into mRNA vaccines like we saw for COVID.
Lots of possibilities. What are the hurdles?
The main challenges are the virus's rapid evolution, its genetic diversity. It changes, making it a moving target for a single vaccine to cover effectively long-term.
Always complicating things. Okay, wrapping up here. Is there any final, maybe unexpected insight about SVV, something people might find surprising?
There is actually something truly fascinating, completely outside of pigs. SVV has shown potent and selective oncolytic properties.
Oncolytic, meaning cancer-killing.
Exactly. Cancer-killing properties against certain human neuroendocrine tumors.
Wow. Human tumors.
Yes. It's actually being evaluated right now in human clinical trials as a potential novel cancer therapy.
That's incredible. A pig virus used to fight human cancer.
It is pretty amazing. And that parallel research, understanding its fundamental biology for cancer therapy, might even give us insights back into the veterinary side eventually. It all connects.
What a journey we've taken today. From what seems like a niche pig virus, we've really uncovered this master of disguise. Seneca Valley virus maybe not always deadly itself, except for those poor piglets, but causing massive disruption, huge economic waves, just because it looks like something worse.
Exactly. And if you think about the big picture, The challenge SVV poses, it really serves as this crucial, ongoing, real-world stress test for the whole swine industry's readiness against all vesicular diseases.
So the fight against SVV strengthens the defenses against FMD.
Absolutely. The lessons learned, the diagnostics developed, the biosecurity that gets tightened because of SVV scares all of that is invaluable in safeguarding the industry against truly devastating threats like foot and mouth. It's a powerful reminder, isn't it? Sometimes the biggest impacts and maybe the most valuable lessons come from the most unexpected places.